Okay, thank you for the presentation, I suppose, because I don't know what you say, because I don't check. Okay, I think that the first question is who Fundación CANNA are. Fundación CANNA is a no-profit organization founded in 2011 by CANNA Corporate, and we are dedicated to research of cannabis plant and its relationship with the human body. And to do that, we have two different ways. The first is provide a comprehensive source of information for cannabis users around the world, sponsor participate in events, conferences, studies, and contribute to the dissemination of scientific education. And another one is, doesn't work, okay, OMS laboratory where it analyzes cannabis and components, as well as carry out different types of chemical analysis in order to the quality and necessity of the market. So according with that, our objectives are two. The first is the scientific study and research of the cannabis plant and its active compounds, its uses and derivatives, in particular with regard with the effect in the human body and mind. And the another one is inform the public and support the international medicinal cannabis movement and dissemination information about the plant, its components, its risks, and its use. Always talking in a language adaptable for dissemination for everybody. Specifically, what we do, we sharing knowledge in a simple way. In our website, you can see a lot of papers, a lot of videos in 8 different languages. We have a YouTube channel when you can see a lot of information, audios, postcards, infographics, very useful for researchers, and we participate in social medias like Facebook, Twitter, Instagram, and LinkedIn. So we sponsor organizations, we sponsor scientific events, we participate in seminars and conferences, and we conduct our own studies and collaborate with another research groups. In the slide, you can see some of the studies that Fundación CANNA have developed by yourself, and in this moment, I want to show you one of them. The title is a study of the CBD content in a variety of commercially available creams. First of all, I need to introduce you a little bit what is the atopic dermatitis. This is the most common chronic inflammatory skin disease. Currently, there are not effective cures, so the unique goal that we have to do is the treatment by reduction and control the symptoms. The different factors that cause this disease are the genetic factor, the environmental weather, and the immunological system of the people. You know perfectly the symptoms, dry skin, erythema, and other ones, and even in chronic lesion, the thinning of the skin may be occur. In this part, I show you the most typical cycle of the disease. Everything starts with the alteration of the protective function of the skin. After that, the presentation of the irritating allergens, the inflammation of the skin, the itching of the skin, and stages. And currently, the standard treatment are based in anti-inflammatory preparation, preparation with the corticosteroids and moisture to reduce the symptoms. In a very extreme case, when the lesion is so big, immunosuppressant and antibiotics could be necessary in order to avoid possible staphylococcal bacteria infection. In this slide, I show you a little bit more the mechanism of this disease. In this part, you can see the two different, here I don't know if you can see, two different parts of the skin is amplified in this part. There are a lot of protein in different colors, in blue colors, and intermediate proteins named theramines that create a lipid barrier that regulate the hydrophilic balance of the skin. So when the damage of the structure of the epidermis is done, in that part, the allergens introduced in the system, they activate the immune cells and produce the inflammation. The epidermis becomes to be thickened, the blue vessel dilates, and appear the vesicles. Alternative treatments are based in the use of cannabis by creams. You know that the cannabis, the phytocannabinoids, are located in the female flowers of the Cannabis sativa plant. And these phytocannabinoids have the capacity to bind to the specific receptor that we have in our body. That receptor, CB1 and CB2, create the endocannabinoid system. CB1 receptor is located more abundantly in the brain and in the main organs of the body, and the CB2 receptor is a little bit more located in immune cells, skin, and bones. So when we apply, for example, with a screen, the phytocannabinoids, that interact with CB2 receptor, and this is a new pathway to treat these kind of diseases like atopic dermatitis. So with this scientific situation, and with the inclusion of by the European Union of Cannabidiol as an ingredient for cosmetic use, various creams claim to contain this cannabinoid, and many consumers are buying this product because of the alleged property attribute of CBD. However, this is very funny because there are no obligation to inform users of whether the cream actually contains CBD, or even to guarantee if the CBD concentration is in the package when the manufacturer declares this content. So, in this study, the CBD content of several commercial available creams was tested by a gas chromatography mass spectrometry. This is an analytical technique. And the results that we obtained with this technology were compared with those declared by the manufacturers. So, to develop the experimental part of this trial, during the year 21, 22, and 23, we acquired creams, 15 creams, that say that contain CBD. Due to the complexity of this sample, I say sample about the creams, was mandatory use gas chromatography technology in order to obtain accuracy results. And was mandatory to use the standard addition method. The standard addition method is based on increase on a no concentration of CBD was added increasing value of no quantity of CBD. That process is named spiking. So the analysts increasing the quantity of the spiking could be extrapolate the unknown quantity of the sample that was not spiked. To do so, 200 milligrams of cream was weighted and diluted with 4 milliliters of acetone. After that, the internal standard was added and sharing. Then the sample was filtering, and the liquid was transferred to a vial and introduced in the autosampler of the machine in order to be measured. In this part of the slide, I describe all the technical data of this instrument We use this very complex name of capillary column. We use helium as carrier gas at this flow rate with a split mode. That means that what part of the sample go outside, but this is also sophisticated. The injector temperature was 250 Celsius degrees, using 1 microliter of volume of the liquid that was in the vial, the temperature of the transfer lines, and we are working in the mass spectrometer using the single ion monitoring mode, exactly with the mass charge of the CBD 231.2. So it's a very precise method. Okay, about the results. In this table, you can see in the first column the quantity of CBD, milligram of CBD per gram of cream. In the second one, milligrams of CBD per container. And in the third one, the milligrams of CBD per container specified by the manufacturer. As you can see, only 6 samples specify the quantity by the manufacturer, and only in one case the quantity is more or less the same that the manufacturer specify, even a little bit more. In the rest, 2 samples contain many lower concentration, around 99% lower, and in the other 3, the numbers are not the same. It's a little bit higher, but around 40, 60, and 67% more or less lower concentration. This is another way to show you the results. Only a 40% of the total samples indicate by the manufacturer the content of the CBD. 60% was not specified, even when the manufacturer specified the content, no complying with the specification, 33%, and only 6% is according with the specification. This is the same results, but in another way to show you. As you can see, this is the level that should be have in the sample. Only one sample have a little bit more quantity, and as I mentioned before, 2 samples have around 99% or even 100% of deviation lower that was specified by the manufacturer, and around the other 3 around 51, 30, and 33 lower concentration. And this is another way to say the same results. In 8 samples, there are no concentration from 0 to 0.07 milligram of CBD per gram of cream, and in the another one from 0.17 to 1.11 milligram per gram of creams. As conclusion, the consumer information of the CBD content of the cream is deficient. Only 40% of the cream disseminated specify it on their packaging. The reliability of this information is also very low, as only one of the 6 creams providing this information containing the amount declared. There is also a lack of precision in the information of CBD concentration in creams, 3 and 6, and it's not specified whether the percentage indicated in the package is expressed in weight or in weight volume. Most notably, however, the CBD is virtually absent in a large number of samples, while in others the concentration is so low that it will be very difficult for CBD provide any kind of effect in the skin. The consumer who buy these creams tend to do so for the added value and potential effect that derive from CBD, but if the cream haven't CBD, it doesn't have sense. The manufacturer should be therefore specify the amount of CBD contained in cream, and its information shall be information that the buyer should be aware of. And it's so important to check the sample content in all the batches. And from me, that's all. If you want, in this QR, you can see all the information, all the studies in Fundación CANNA website. And if you have other question or information, you can send us by email everything that you want.